So we left off in the last video quickly just going over what a prokaryote is, a basic cell. It doesn't have a nucleus, it's really tiny, and we kind of think about them as bacteria and archaea, which you'll hear more about in microbiology. These two cells over here, these are called eukaryote cells. You can notice a couple of things. These guys are a lot more complex. They have a lot more stuff going inside the cells. This one over here is a plant cell, and this one over here is a animal cell, like us. So they're going to have a whole bunch of different components that they share and do the same function in each of these. Some will be unique just to one structure, like this guy is going to be a chloroplast. We're not going to really discuss them too much until we get to our later chapters. The first thing that these cells have in common, so this is going to be prokaryotes, eukaryotes, and plants, will be a cell membrane. This will be something that keeps stuff from the outside from coming in and inside coming out. We've talked about this a little bit already in chapter 3, but basically they kind of make it so they're semi-permeable. Some things can come in, some things can go out, and they basically use osmosis, diffusion, or if they have to pump something the opposite direction, active transport, to get stuff in and out of the cell. But the organelles that we're really going to focus in on for this one is going to be the endoplasmic reticulum, nucleus, ribosome, and the Golgi apparatus. I'm going to keep referring back to this photo and then moving forward, but basically what happens is we have the nucleus, which we've kind of already talked about. That's where the DNA is. Outside of that, we have these other structures that help support how the cell works and moves. The nucleus is going to be, I like to think about it like the CPU of the cell. It's where the DNA is, and the DNA is inside this. The nucleus basically protects the DNA. It's kind of like a safe, and only few things could come in and out of this, and it's very regulated. The kind of stuff that can come out is this thing called RNA, which we're going to talk about soon. It's just like a message from inside the cell to outside the cell. And a few things could come in like hormones, and we'll talk more about that in Bio 202. These little things that are sort of studded right over here, that's going to lead us to our next thing. That's going to be our ribosome. The ribosome's function is to make protein from a specific type of RNA, which we're going to discuss in the future. It's made up of this instructions that's basically going to give the cell the ability to make a specific protein. So when I think about this system, I think about this making basically a recipe, and that recipe gets sent out to these little bumps called the ribosome, and those ribosomes take that recipe and make it into a product. The endomembrane system, there's actually two of them, and basically there's a rough ER and a smooth ER, and basically what they are designed to do is functions to flow one organelle into the other, and they help with the regulation of making proteins. So the endoplasmic reticulum, there's two of them. There's the rough ER, and it's called rough because there's ribosomes all over it. And it's designed for protein synthesis. And then the other side of that, and I'll show you a picture of it in a second, is called the smooth ER. It has no ribosomes, and that's why it's called the smooth ER. There's no proteins that are being made here, but what happens in this area is going to be lipid synthesis. Remember those lipids on the outside of the cell? And metabolism. Some metabolism occurs here. Detoxification. And these vesicle formations. And if we have something really big that we've got to move outside of the cell, we use these vesicles to move them from one side of the cell to the next. So we said there's a smooth and a rough, and just to give you orientation, this would be the nucleus, and then if I blew this area up, you'd see that there's a rough ER. The rough ER is the one with all the little dots. Those dots represent the ribosome. the factory that basically makes those proteins from the instructions that come from the nucleus. And then over here, we have a smoother area, and we've just depicted them in two different colors, so you can see that one was rough and one was smooth, but this is where that lipid synthesis occurs in those vesicles and detoxification. So if this was the nucleus, you could see a ribosome, and a ribosome is going to make the proteins. And if you recall back from chapter 3, a protein is basically just amino acids put together in the right order, and eventually they start to fold in a specific way. Remember those primary, secondary, tertiary, quaternary structures? Well, this is the process of its folding, and let's say this is the finished product, the quaternary structure. You'll notice that, number one, there's these little glycoproteins on the outside of the protein. And what they're designed to do is they're kind of like a zip code. They're telling the cell where to deliver it. And then what happens is this smooth ER, let's say this is the smooth ER, is going to go ahead and make this vesicle and deliver it based off of where it's going to go. The sugars tell it where to go, and this is the protein that needs to go either outside the cell or somewhere inside the cell, and it's going to get delivered. If we continue, we have the Golgi apparatus. The Golgi apparatus is going to be, and I'll show you a quick picture of it. So you can see here's the nucleus, here's the rough ER and the smooth ER. Things usually get delivered right to the Golgi apparatus. And the Golgi apparatus functions to modify these proteins. So it'll take protein A and B and C and D and stick them together so they function. And then what it does is it sends it wherever it needs to go. So it's going to send it either outside the cell, and that's called secretion, or it's going to send it somewhere else in the cell. So it's going to send it from one part of the cell to the other. And so when I think about the Golgi apparatus, I think about the UPS system of the cell. So things can get delivered inside the cell, or things can go outside the cell and get delivered to somewhere else. So let's say if you had to send a message to the pancreas, or you just have to send a message to the kidneys or the liver, this is going to be sent through this method. Big picture, nucleus, inside there's DNA. The message is going to go to the rough ER, and it'll get made into those proteins. Eventually what will happen is we'll go to the smooth ER. Smooth ER will deliver it, or temporarily take it in these vesicles, to the Golgi apparatus. That's what this green thing is. And what will happen here is we could add some of those proteins together to be the final product, or when the time is right, we're basically going to push it. So this would be outside the cell. We could secrete it outside the cell, or we could move it somewhere in the cell. So for instance, we could package it and move it somewhere else in the cell. So this is going to be the UPS system. This is going to be the manufacturing area. And of course, this is going to be that CPU. And then we have some things that are involved in breaking and recycling things down. So when I think about your house, let's say, because a cell works very similar to that, you have two garbage cans. You have one that's designed for recycling, and you have one that's designed just for trash. The lysosome is going to be the one that is going to basically take things and break them down. So let's say you have some proteins you don't want to use anymore, you have some fats or whatever that you want to break down and reuse. That's going to go to the lysosome. So this is going to be like a recycling system. Where the peroxisome is this area that if we don't know what it is, or we think it might be harmful, we put it inside here, and it kills it. And you might recognize some of this word from peroxide. Well, you know, peroxide is something that if you hit it with bacteria or something like that, it bubbles and kills it. Well, that's where this comes from. So this is going to be like the incinerator of the cell. The garbage goes here. And this is where the recycling stuff comes from. And this area is only found in a few organisms, but all plants. And vacuoles are basically just going to be an area where we can store water. So in plants, if you've ever seen a plant that's standing up and then you forget to water it for a few days, it gets kind of droopy. These vacuoles sort of lose water. And then as soon as you hit it with water, within maybe 10 to 20 minutes, the plant goes standing back up. And it's because these vacuoles went from being shriveled and not full to completely filled, and that's what's giving that plant that rigidness to stand up. So here we are, the lysosome. It's got some digestive enzymes in there, and that's the recycler. So it's always going to be able to sort of break things down and recycle them. The vacuole, again, so here's a organism that has a vacuole. Not everything does have it in. So you can see it's a big structure right inside. It's filled with water. So it's usually a water reserve for lots of things. That's why, like here with this plant, you could see that this is the vacuole. And it's huge, so the plant doesn't have to have water 24/7. It can just keep tapping into its vacuole and use it. But keep in mind, only plants and some organisms have it. Most eukaryotes don't. And peroxisomes, they function by breaking down fatty acids, detoxifying alcohol, and really destroying things. If we look at the summary, you could see that we've talked about the nucleus, we've talked about the rough ER and the smooth ER, the Golgi apparatus, and then lysosomes, and somewhere around here there's a peroxisome, and this would be the outside of the cell. So we've talked about a couple of structures. In our next one, we'll talk about the energy of the cell.